A warm welcome, dear children. We will start with the experiment, paper chromatography experiment, and this is one of the experiments for your practical work, and the objective is to separate and study the plant pigments by paper chromatography, right? That means to separate and study the plant pigments by paper chromatography. Now before we start with the experiment or the practical work, let us understand what chromatography is. Right, this is the figure that you can see, that there is a beaker, and in the beaker is the chromatography paper. Right, so chromatography is derived, the word chromatography is derived from a Greek word chroma, which means color, and graphe is writing. So this is a technique used to separate molecules of different substances present together in a solution according to the rate they travel along the chromatographic paper or a column of absorbent material. So here you can see the chromatography paper, or it is an absorbent material, and you can see how the substance has traveled all along the paper, and you can see the visible colors that are there. Now such a preparation is called chromatogram. The solution containing different substances is applied to the chromatogram and is allowed to flow. So in the beaker you can see that there is some liquid also present. So here is the chromatography, the proper diagram that you can see over here, how it is done. Right, so the molecules of different substances travel through the medium at different rates, right, and that is how you can see that since they travel at different rates, so they get separated. You can see the solvent is here, right, the spot is there, that means the whatever substance has to be tested. Now here we will be testing the plant pigments, right, so where ink spot is written, there the plant pigment will be put, right, and then you will see that it would travel along, and it would occupy, it will get separated and would occupy different place on the chromatographic paper. Right, so the molecules of different substances, they travel through the medium at different rates and they get separated, and this is how you can see how it gets separated. All along it travels, right. Depending upon the medium of the chromatogram, chromatography is of 4 types. Right, so these 4 types are paper chromatography, column chromatography, thin layered chromatography, and partition chromatography. What we will do is paper chromatography. So in paper chromatography, the mixture containing different compounds is spotted onto a chromatographic paper, right. So we can take this to be the white strip to be the chromatographic paper, which is the filter paper shown over here, right, and then a solvent is allowed to flow along the paper sheet. You can see the solvent that is shown over here, it flows along the paper sheet, that is the chromatographic paper. So it is spotted onto a chromatographic paper and it is dried. Then the solvent is allowed to flow along the paper sheet either by gravity or by capillary attraction. As the solvent flows through the paper, it spreads the different compounds of the mixture into various distances. You can see how the compound has spread into various distances, right. So that is how it is done, and then the paper is dried. The position of different compounds are visualized either directly, such as pigments, which we will see directly, or by suitable staining reactions. Right, so we will be able to see the different pigments as shown over here. So different plant pigments, that is the photosynthetic pigments, can be separated by using the technique of paper chromatography. So now in the next video we will talk about the requirements of paper chromatography. Again I would repeat that the objective is to separate, or the aim is to separate and study the plant pigments by paper chromatography.